Hi, my name is Elizabeth Saunders and my project is titled Soccer Cleats vs. Household Cleaning Products. I decided to research the most effective way to clean my soccer cleats because as a soccer player myself, I've struggled to find ways to eliminate the odor, sweat, rain, mud, and other substances found on my soccer cleats for both my personal hygiene as well as to eliminate cross-contamination between different environments that I play in. To begin with, I looked at the research of others in order to decide how I could contribute to the scientific field, as well as to determine what I wanted to specifically look into. So I looked at 3 specific studies and focused on those, while I also looked at many others. So first, a study at Penn State showed that Clorox wipes are truly 99.99% effective, but they are only that effective if they are used once. So I used this in my own research because when I used Clorox wipes, I used each Clorox wipe once, and then I threw it away and used another Clorox wipe. A study at the University of Arizona showed that 90 to 99% of the bacteria that is on bathroom floors is transferred to the bottom of your shoes. And so this is very important in my research because it tied directly to the bacteria that is transferred onto your bacteria cleats, and then also the best way to transfer that bacteria back off your cleats. And finally, a study at Sierra College showed 13 different colonies on their home college turf were found that were not native to their college turf, which was really important and showed me how much cross-contamination there really is between areas. And if you want to keep a different environment ecosystem in their purest form, that you need to clean your cleats before you play in different areas. And so that tied into what I wanted to do and also my variables. So my independent variables that I changed were the Clorox wipes, which are found in many households, and the active ingredient is bleach, and Norwick Sports Enzyme Spray, which is a bit less common and it's more of a sell-at-home product, and you can also find it on Amazon. And it is advertised as an odor eliminator, but it is organic, and it uses organic enzymes and microbes to kill bacteria. And so it's a lot different than the bleach found in Clorox wipes. And so I thought by comparing 2 very different things, I would get to see how the results compared. My dependent variable in my field test were the number of bacterial colonies found, and in the sensitivity test, it was how large the clear zones were. And I had many control variables, including using the same trypticase soy agar plates, always keeping the incubator at 37 degrees Celsius, and putting each TSA plate in for 24 hours. I also used the same heat fixing and gram staining methods and procedures, and I also had a confirmation slide for both my right cleat after Sports Enzyme Spray and left cleat after Clorox wipes. So now moving into my method and procedure. Originally, I first practiced in both cleats, and I then took a swab from both cleats using a sterile cotton swab. I then placed those cotton swabs into plastic bags. In my left cleat, I wiped with 3 different Clorox wipes. Each wipe was used once and then disposed of. And I took a swab from my left cleat immediately after that. And in my right cleat, after I took my original swab, I sprayed with the Sports Enzyme Spray. However, I waited 30 minutes because the area was still wet from the Sports Enzyme Spray. And so that is one thing I would change in the future. I would also wait 30 minutes before I took the second swab for my right cleat. So after I took my right cleat, that was also placed in a bag. After I had all 4 sterile cotton swabs, they were all rubbed on their own individual trypticase soy agar plates. Those plates were then placed in the incubator for 24 hours at 37 degrees Celsius. Then when they came out, they looked like this, and I circled and identified and observed each different type of colony found on the TSA plates. And for each circle and number, it is a different colony, and each colony then got its own slide and was heat fixed, gram stained, and observed under the microscope. So these observations themselves, first we had Corynebacterium pseudoephedrine, Streptococcus faecalis, Staphylococcus aureus, and finally Bacillus megaterium. And so those were found each from, these are all one slide that was observed under the microscope from one of these colonies in their category. And I then, after observing and identifying, after I looked at all of them under the microscope, I used all the information from the morphology chart as well as from the pictures from under the microscope to create, to try and identify each bacteria. So this was the morphology chart I used originally. However, I then translated that morphology chart into these results, which we'll go over later because I believe they're easier to understand. And this morphology chart will also be in the Science Fair website if I can successfully upload it. So, going to the sensitivity test, now that we're back to the method. In my sensitivity test, I soaked 3 discs in the liquid from the Clorox wipes and 3 discs in the liquid from the Norwex sports enzyme spray. I then used Bacillus cereus as my bacteria sample because it was found in both the right cleat after and left cleat after samples, meaning it was found in both the right cleat and left cleat, and it was not eliminated by either directly originally. And so I wanted to see which one, which substance was more successful in eliminating the bacteria in the sensitivity test. So on the left side, I took the Bacillus cereus from the left cleat after Clorox wipes and rubbed from the edge to the middle. And on the right side, I took from the right cleat after Norwex sports enzyme spray and rubbed from the right to the middle. I then placed the discs, respectively, the Clorox on the left, the Norwex enzyme spray on the right. They were evenly-ish placed. There was some error because of the accuracy with the materials we were given. And then I measured the average clear zones, which we'll go over in results. So now for results for the direct field test, there were 7 colonies found in both the right cleat and left cleat original cleats and original samples. However, after the Clorox wipes and Norwex enzyme spray were used, there were 5 colonies in the right cleat after the sports enzyme spray and 4 colonies in the left cleat after the Clorox wipes. Another major difference was that in the right cleat there were more irregular, whereas there were more circular in the left cleat. And there's about a 50-50 ratio for gram-positive cocci and gram-positive bacilli. And there's also, however more gram-positive cocci were removed than gram-positive bacilli. And there's also about a 50-50 ratio for the pigmented and non-pigmented. And there was also human error evident because if we look again at the circular and irregular, for the right cleat there were originally 7 circular, and then after it was treated with the sports enzyme spray there were 2 irregularly shaped, and so that shows how there was likely some human error because there were likely no new bacteria. However, it was just, there could have been new bacteria, or I could have just said something was circular where I might have already said it was irregular. And so that was just some human error that I would look to try and eliminate if I were to do this project again. And for the results of the sensitivity test, the Clorox wipes had a 13 millimeter clear zone, whereas the Norwex enzyme spray had an average of a 7 millimeter clear zone. And so the Clorox wipes were more effective in both the sensitivity test and the direct field test, where they eliminated one more kind of bacteria. So if we analyze and draw some conclusions overall, the clear zone was 85.71% larger with the Clorox wipes, and the Clorox eliminated 14% more bacteria colonies in the field test. And additionally, the results are not entirely reproducible because although the Clorox wipes would likely perform better than sports enzyme spray if you were to perform this on your own cleats at home, because everyone practices and plays in different environments and has different kinds of bacteria on their cleats, it is not entirely reproducible. And if I were to do this test again, I would test the soils that I had played in the different areas to see the transfer rate between the grounds that I had played in and my cleats themselves to see where that bacteria had come from and if I had any cross-contamination on my cleats at the time. I would also test more substances to see the most effective way of killing the bacteria, so not just Clorox wipes and the sports enzyme spray. And finally, I would try and reduce human error in my morphology chart, and then just throughout by using more trials. Thank you.